Hi, I'd like to thank you for this kind invitation to speak at this important program and to the MPS community for hosting this collaborative conversation. My name is Robin LeWinter, and I am the JCR USA Medical Affairs and Medical Communications Lead. And I'm looking forward to sharing out more information about JCR, who we are as a company, and discussing our exciting platform designed to deliver enzyme across the blood-brain barrier. So before I get into details about our platform and therapy pipeline focused on MPS, I'd like to please allow me to introduce a little bit about our company. JCR is a Japanese pharmaceutical company founded in 1975. Until recently, it's been predominantly a domestic company with multiple marketed products in Japan. The 49 years of its history, we focused on manufacturing, research, and development of process technologies and new treatment options, especially for orphan and rare diseases. As you can see from this slide, we're headquartered in Kobe, Japan, and we have a number of other offices distributed in the rest of Japan. Importantly, we have 2 research centers focused on discovery and proof of concept, and 5 manufacturing sites that sustain our products marketed in Japan and for our clinical trials, which highlight our focus on development. More recently, JCR has expanded our global footprint, and we're now reaching out to patients beyond Japan. And today we have a presence in the USA, Brazil, and the Netherlands to sustain global clinical development for our programs for rare diseases. So I'm now going to share the basic premise of J-brain cargo technology and why we're so excited to explore its potential in mucopolysaccharidoses. So as we all know, MPS is a lysosomal storage disorder. Sorry, I jumped ahead. It's a lysosomal storage disorder, and while enzyme replacement therapy seems like an obvious solution, enzyme replacement therapy has been unable to cross the blood-brain barrier and to treat the neuronopathic components of the disease. The blood-brain barrier is a very selective filter that blocks the passage of many substances from the blood to the brain, including pathogens, but importantly and unfortunately, enzyme replacement therapy. So JCR has created a technology we call J-brain cargo that allows effector molecules like enzyme replacement therapy to enter the brain by taking advantage of the body's carefully regulated blood-brain barrier to deliver enzyme to the brain using the receptor-mediated transcytosis that's been talked about today. Well, how does it work? So all cells require iron for oxygen exchange. And iron is safely carried across the blood-brain barrier using a specialized glycoprotein called transferrin. As the cells in the brain need iron to function, your body has created a mechanism to transport transferrin across the blood-brain barrier. Transferrin binds to a specialized receptor, cleverly called the transferrin receptor, on one side, and it's encapsulated and carried across to the other side of the blood-brain barrier where it's released into the parenchyma of the brain and is now available for use by cells in the brain. J-brain cargo has hijacked this process. So we've developed a technique to fuse an antibody to the transferrin receptor to an enzyme. And if you remember, an antibody is like a puzzle piece which can connect to its antigen. So in this case, the transferrin receptor exactly matches the antigen on the J-brain cargo. And it, just like with transferrin, it takes it across the blood-brain barrier and into the brain. It's key to point out that this missing enzyme is attached to the antibody. So the missing enzyme is transported into the brain and is now available for use in the CNS. Our drugs are administered IV into the blood, and they're distributed throughout the body as well. So as I've discussed, J-brain cargo is able to cross the blood-brain barrier and enter the brain, but it's also important to note that since it's administered intravenously, it's readily available for uptake in all somatic organs via the mannose-6-phosphate receptor and transferrin receptors on all cells. Therefore, we postulate that this treatment provides benefit both in the central nervous system, but also on somatic signs and symptoms. And we all know MPS affects both the somatic body, and the central nervous system. So now I'd like to share with you data on JR141, also called Pabinifest alpha, it's a hard word to say, which is commercially available in Japan, called Cargo. So this is a really complicated slide. I'm just going to touch on the highlights here. But first, we needed to show that it worked preclinically. And so in the top left, you can see we've using a mouse model of mice who have MPS II. The mice were infused with JR141. Enzyme replacement therapy or placebo control. And if you look in the top left, all the way on the left, you'll see that the wild type mice had no heparan sulfate in their serum, whereas mice who had the mouse model of the disease had quite a lot of heparan sulfate. And when they were treated with JR141 or enzyme replacement therapy, the heparan sulfate levels went down to close to normal levels. Now on the top right, we also looked at the CSF. And what you can see is that mice that on the blue on the right, the mice that were the diseased mice treated with enzyme replacement therapy had no decrease of the heparan sulfate levels, whereas the mice treated with JR141 did have decreases in heparan sulfate levels. In the bottom left, you can see this reflected in behavior. I won't get into this in great detail, but essentially there's a preservation of spatial learning in the mice who were treated. Now, the bottom right is some new data that was recently presented in Japan. As Dr. Pinedo so nicely described earlier, there is vision issues in patients with MPS. And so we looked at the retina in these mice, and what we saw is that JR141 also reduced the heparan sulfate levels in the retina of these mice. So this led us to treat patients. These are the studies that led to approval of this drug in Japan. And on the left, you can see that there were patients were treated with JR141 for about 12 weeks, and their levels went down. They were then removed from the drug for a year, and there was a rebound in the levels, which then, when the patients were put back on the drug, their CSF heparan sulfate levels again went down very low. And on the right, we are showing the somatic effects on JR141 by looking at spleen and liver volume, and you'll note that they are equivalent to enzyme replacement therapy. The safety profile of JR141 has been studied for 8 years now and evaluated in almost 100 patients in clinical studies and 50 post-marketing, and there have been no significant findings. Some more safety data. But all of this data together, we are so proud that these studies have led to the development and approval of Cargo in Japan in 2021, which has been shown in clinical trials to reduce the production of heparan sulfate in the brains of patients with MPS II. Now, one of the nice things about having an approved drug is we can do some of these post-marketing studies that Dr. Lau mentioned and focus on other aspects of the disease in patients who have been treated. And so here we show interviews with patients and caregivers who have received our drug for about 3.5 years. With most of these caregivers are reporting a trend towards improvement in multiple aspects of their children's disease, including cognitive improvement in language, concentration, and self-control. And some are even reporting improvements in the whole family's quality of life. So the approval of Cargo in Japan marked a new chapter for JCR, and we're now proceeding to develop additional treatment using our J-Brain Cargo technology. So this is our pipeline. And I'm going to just real quickly speak to those in the blue square. So we are now working to expand our work in JR141 beyond Japan, and we're in the middle of a global phase 3 study designed to demonstrate the safety and efficacy of JR141 on CNS and somatic signs and symptoms called the Starlight study. More details of it can be found on clinicaltrials.gov and on the official website listed here. And just for the purposes of this audience, there are 6 sites open in the US, which are distributed around the United States. JR171 is, I'd like to segue into a clinical trial we've got going on for JR171 and MPS1, where we're in the midst of a phase 1/2 study designed to determine long-term safety and dosing in this patient population. The initial 12-week study is complete, and we're now in the midst of a 143-week follow-up. Results from this study at 1 year were presented at World last year, where you can see that there was a decrease in heparan sulfate in most subjects, all subjects actually, and the overall safety profile is comparable to somatic enzyme replacement therapy. Lastly, we have 2 earlier phase trials going on for MPS IIIA and MPS IIIB. We've got 2 phase 1 or a phase 1/2 study initiated in MPS IIIA, and we've recently dosed our first patient in Japan for JR446 for MPS IIIB. So thank you for your attention.